Okay, please, on your lab report, write either Tuesday or Wednesday and bring them forth and make sure I check you off, all right? Don't all rush me at once. All right, Wednesday, Leah. Okay, you're the step. Okay. Donna, you're in Tuesday. Okay. On your Tuesday. Let's actually open this one. Okay. Matt, Tuesday. Okay. All right. Cindy, Tuesday. Okay. All right. Caroline, you're Tuesday. Okay. Where does it say Tuesday, Wednesday?
Tuesday, Wednesday, no, oh, it's just the 1B part. It's Wednesday.
Wednesday, all right. Hey, Bruno. All right, gotcha. All right, Katrina, you're Tuesday. Okay. Wednesday.
Not really. I never have everywhere.
Okay. Wednesday. Okay. Tomorrow, your Tuesday. Okay. Wednesday. Victoria. Okay.
I'm in Wednesday, but I didn't graduate this week. And it fell apart, so there's one.
I applaud resourcefulness. There we go. Sharif, you're Tuesday. Okay. Christina, you're Wednesday or Tuesday? Tuesday, Tony. Did you write it on here? No. Okay. Write Tuesday or Wednesday, please. Big, so I can see it. Okay. Are you getting these all mixed up? Yes. Tuesday, Tuesday. Tuesday, Justin. Tuesday, last name. Okay. All right. Hillary, you're Wednesday. Okay. Andrew, Wednesday. Okay. Off Wednesday. Okay. Wednesday. Okay. All right. Lynn, Wednesday? Wednesday. Okay. All right. Tuesday or Wednesday? I just got to read carefully. All right. All right. All right. Wednesday. Okay. All right. Wednesday. Any more papers? Thanks. TAs are not here? All right, I gotta start the lecture. David, I'm going to have you check these gentlemen in and subsequent ones. There's check-in sheets here, and there really is a Tuesday and a Wednesday stack. All right, thanks. Okay, for this lecture, what I'm going to do is I am going to show you what motivated the study that you are replicating and extending, and give you some intellectual context into which it fit. You don't necessarily, and I think you probably shouldn't, review all these studies in your lab report. Okay, so for your lab report, I'm going to ask for a more bare bones, just why the heck are you doing this, and defining things in your introduction. Don't go into some elaborate review of all these studies. But this will give you a sense of why was this study ever done, and why was it at least a little bit interesting. Everybody who's coming in now, please go see David and get your papers checked in. All right, so this is actually the story of how I became a hormonal heretic and backed away from the ideas that are presented in all the textbooks, at least a little bit. And the idea to which I finally had to have some exceptions is one that I firmly believed in, because my professors had taught it to me and it actually accounted for a lot of data, and that's this dogma that I talked about on Monday, that sexual differentiation of the brain is driven exclusively by gonadal steroids. Remember last time we talked about the genotype determines the gonad, and after that it's the secretions of the gonad that determine sexual differentiation, including of the brain and, of course, as a consequence, behavior. So that's the dogma, and we talked last Monday about some studies that were motivated by this central dogma, and to remind you, we talked about a study in which we looked at which hormone masculinizes the song system best. And so to explore this, what we're doing in these studies oftentimes is giving hormones or manipulating hormones in females, and then by inference figuring that that's what happens naturally in males. Okay? And the study that I talked about on Monday, we looked at a whole series of hormones. We looked at progesterone, we looked at androstenedione, which is an androgen, we looked at testosterone, which is an androgen, and we also looked at estradiol, which is the estrogen that is the most potent. And I should hasten to point out here in this slide, there's an arrow going from testosterone to estradiol, and all these arrows in this whole diagram represent enzymes. And so there is an enzyme that changes testosterone to estradiol. This enzyme is known as aromatase. And why is it called that? See this ring right here and the testosterone molecule? So what happens is this enzyme breaks this double bond here with the oxygen, turns it into a single bond, and then turns this into an aromatic ring. This ought to come back to you from all that wonderful organic chemistry that you loved, right? So that's why it's called aromatase, okay? So hang on to that idea about aromatase. It's going to actually figure largely in this whole story as it develops. So at any rate, and also you should remember, testosterone can get converted into estradiol, and it's a one-way reaction. Testosterone to estradiol, doesn't go back. And the other thing that you should know is that there are some neurons in the brain that are really good, that express this aromatase enzyme, and so can grab androgens like testosterone and convert them to estrogens. Okay, so there are neurons in the brain. We know this for sure in mammals and birds that express this enzyme aromatase and can convert testosterone to estrogen. So last time, on Monday, I talked about the fact that when we compared, what we have here is the RA volume. You've now seen RA in the lab. We had a group of males that were controls and all these different groups of females, and the blank ones were the females that got no hormone. Otherwise, all these different groups got equal doses of progesterone, androstenedione, testosterone, and estradiol. And did we get a sex difference here? Let's talk about how to read these kind of graphs. Did we get a sex difference? Why do you tell me this? Yes. So from this graph, how can you figure out that I got a sex difference? All right, blank, and what explicitly? What little detail compared to males, and what little detail on this graph? The little asterisk, thank you. All right, so this little asterisk, I'm trying to indicate here, this little asterisk relative to this group, that this little asterisk shows that this group relative to this group is significantly different. Okay, so yeah, got a sex difference. Okay, and how about estradiol? Did we get an effect of estradiol? Yes, so again, we have this little asterisk relative to blanks. So estradiol masculinized. How about testosterone? Did we get an effect of testosterone? Significant, yes. So we got a significant effect relative to blanks. So that's how you interpret these kind of graphs. But the one sort of odd thing, actually it's not odd, that estrogen turned out to masculinize better than testosterone. Why is that? As I just mentioned, there are neurons that can convert testosterone into estradiol in the brain. And so we think that what happens, at least in mammals, is the testosterone gets delivered to the brain and the neurons flip it into estradiol, and it's estradiol that is the molecule that kicks off masculinization. Okay? Yeah? This is a developmental thing to change in the organizational circuit, but this also happens in adulthood. Okay, so literally your neurons and my neurons are grabbing some testosterone and flipping it into estradiol, and then it's having some biological effects within the neuron. The delivery molecule to the neurons, however, for us is estradiol. Or, excuse me, it's testosterone. Okay, so estradiol masculinized better than testosterone. That actually turns out not to be so weird in studies with mammals where they've done nose-to-nose comparisons, equal doses, in other words, of estradiol and testosterone. Estradiol masculinizes behavior and brain better than testosterone. Okay? The reason why is testosterone, as I mentioned, gets metabolized to estradiol, but it also gets metabolized to other things. Okay? So the conversion to estradiol from testosterone is not 100% efficient. All right, so an equal dose of estradiol and testosterone wouldn't necessarily mean that these guys that got, these actually girls that got testosterone would have had an equivalent amount of estradiol ultimately. Does that make sense? They'd in fact have less. All right. So is this a weird bird thing? And I've already said no, and I'm going to say it works the same as in most mammals, and I should append that to say rodents. Okay, so most of this stuff has been done on rodents, and maybe by the time we get done with this module, I'll have some time to talk a little bit about what we think might even be true in humans, okay? But we'll see how time works out. Okay, so there was another thing to note about this that we didn't even see in the data at the time, and that was that her brain only got half masculinized by the estradiol treatment and not completely sex reversed. And she counters that this was the most sex reversed zebrafinch brain in history. But if we look at the data, we can see, okay, here's the male and here's the female treated with estradiol, and relative to the male, it only half masculinized. Now, we should have been troubled at that point. We weren't. and I'll explain why. But we should have been troubled because the dogma is, remember the dogma is that gonadal steroids are exclusively responsible for masculinizing the brain, okay? So if that's true, how come we can't completely sex reverse this brain with gonadal steroids? We didn't worry about it at the time because there's a lot of reasons why that might not be true with our experiment, okay? So recall that in order to get this kind of organizational effect, one of the things is that there's a critical period, right, or a sensitive period. And so we don't really know in what biological mechanisms, to this day we don't know what biological mechanisms determine this sensitive period. So it actually could have been ramping down as we gave this estradiol. So maybe the sensitivity to the system was closing down as we gave the dose, okay? So, yeah. Actually, what I'm meaning here is actually the sensitivity of the brain to these steroid hormones. So in at least rodents and in the zebra finches, there are a few days in early life that you can get these hormones to cause changes in the brain. And if you wait beyond those few days, in just post-hatch or post-birth, they won't work. I mean, you can give like a pound of estradiol and you cannot get the brain to sex reverse. Like 20 days is the limit for zebra finches. 20 days after hatching, it doesn't matter what dose you give. You cannot get that system to masculinize. We don't know yet why that closes down. I am betting a whole lot it's on methylation of DNA will ultimately be the answer. But for reasons that I am not clear about, nobody's really looking about why these periods close down. So I suspect for some reason this DNA gets methylated, can't be transcribed anymore, but we really don't know why and how that methylation works. Okay, so one of the reasons we didn't worry about is, well, okay, we don't really know that when the critical period is open and how ramped down it is or what. And the other problem is you always got to worry about dose. You know, maybe if I had given a bigger dose, could have done it. All right, so I think I gave 75 micrograms in this study. Man, maybe gave a couple hundred, would have worked. All right, so we didn't worry about it at the time, and this study alone wouldn't suggest that there's anything wrong with the dogma. Okay, but as we weave this study in with others, we're going to see this dogma is going to start to unravel a little bit. So as I got into this, one of the questions that I thought begged answering, and I still feel like begs for some answers, is how the heck, what are the mechanisms that estradiol kicks off in this female to masculinize her brain to act, to look and actually act? Females will sing, by the way, if you masculinize their brain. They give kind of a crummy song, but they will sing. So what is it that happens? What does estradiol kick off that makes their brain masculinize? And so I went back and I thought about in the literature what we see in terms of sex differences in the neural song system. And as you've actually already seen in lab, you've seen the volumes of the song regions. You were blind, but some of those had really different volumes, right? And one was a male and one was a control female. And by the way, the data so far look great. The numbers of neurons, we're not getting down to that level in lab, but if you went and counted all the neurons in the song system, you would see that males have more. The size of the neurons are bigger in males. And there's one special thing. Males express androgen receptors in their song system neurons to a greater degree than females do. So they can accumulate androgens in their song system, whereas females can't so much. So that's what I was going to key on here. It's like, well, there is this sex difference, and so how does estradiol play into creating these sex differences? And here again is a schematic of the birdsong system. This is kind of a semi-sagittal. Anterior is over here, and posterior is this direction. Here's the cerebellum. And if you give estradiol to these little baby females, what happens is androgen receptors start popping up in the song system where normally in development they would not occur. All right, so I knew from another study that if you gave estradiol, androgen receptors get upregulated in the song system. So that led me to hypothesize, as does this little zebrafinch here, that what happens is estradiol is actually paving the way for androgens to act in the song systems, and maybe it's androgens that are actually doing the masculinization in this system. Does that make sense? Okay. And that would be especially nice and true if the little girl zebrafinches already had high levels of androgens when they're babies, and it turns out they did. And as she recalls, she had a lot of androgens when she was younger. We don't know why they have a lot of androgens when they're little babies, but they do. So they've got the substrate. If they then had the receptor, then the androgens could act. All right? So my idea was, oh, okay, estradiol may cause the androgen receptors to upregulate, and it's androgens that are responsible. So I did this study. And what this study, I got 4 groups. They were all females. And I did what's called a 2-by-2 design. Okay, so in our design, in our experiment, how many factors do we have? Bless you. Anybody remember? Oh, all those figuring out molecules. 1 factor, how many levels? 5. Okay. This is a different kind of design. This has got 2 factors, each with 2 levels. Okay. And so what I did was I gave estradiol or no estradiol. Okay. So that's one of my factors, estradiol or none. Okay. And then what I did was I was interested in blocking the effects of androgens. So there's a drug that binds to the androgen receptor called flutamide, and what flutamide does is bind to the androgen receptor and keep the androgens from getting on that receptor so they can't act. All right, and flutamide by itself does not act like an androgen. Okay, so this is what we call a 2, because we have this factor with 2 levels by 2 design. Okay, so one group gets estradiol plus the flutamide. This group gets a blank for the estradiol, but it gets flutamide. This group got the estradiol alone plus a blank. All right, and then this group got a blank for the estradiol and got the flutamide. See how this works? Okay. So again, we gave these treatments to babies, waited till they were adults to take their brains, and again to talk about how flutamide works, I pulled this in from my first lecture, and what happens with steroid hormones, they go through the membrane because they're lipophilic, and then if there are receptors in the cell, there will be some protein receptors, usually in the unbound state. They hang out in the cytoplasm. Okay, so there's some evidence now that there might be some membrane steroid receptors, but the classic ones that we know about the best hang out in the cytoplasm. And when they bind the steroid hormone, they go into the nucleus and bind to the DNA and then kick off transcription. What flutamide does is it binds to this androgen receptor and so keeps the androgens off it so they can't act, okay? And so they just get stuck in that state and they don't bind to the DNA, all right? So flutamide is what we call an anti-androgen. It is used clinically in really fierce cases of prostate cancer where it keeps coming back and keeps coming back and keeps coming back. They'll give flutamide, okay, to shut down the androgen actions. Okay, see how it came out with the old zebrafinches. Okay, so here we've got HBC volume as a function of my different groups. Here, this is the group that got estradiol alone. And so did we get a masculinizing effect of estradiol? Okay, so why do you tell me these things? Sorry, I love drill. What about this graph tells you that you got a masculinizing effect of estradiol? The asterisk, and then it's relative to this group. This is the control that got blank and blank. Okay, so wait, there should be a blank and blank. Ooh, messed up back here. Take that F off. There should be blank and blank. Okay. So they got no hormone, no blockers. So relative to these guys, yes, there is there's a masculinizing effect of estradiol. Now, when we gave the estradiol in conjunction with flutamide, what happened? No masculinization, completely blocked it. Okay, so one of the things you got to worry about is, oh, maybe flutamide kills off neurons. What's it do all by itself? So we put in the flutamide alone, didn't have any effect at all relative to controls. What's this NS stand for, by the way? Not significant, right? Okay. So blocking androgen action completely blocks the estrogen-induced masculinization. I was pretty jazzed about this paper. And so were some of the students that helped me. So just to give them good credit, Janet Lee and Mary Ellen McCormick are off being doctors now. Kay Yang Stainer was an undergraduate here, and now I still run into her with her kids. She still lives in the area once in a while. Okay, so the conclusion is estrogen's masculinizing effect is mediated by androgen receptors. Everyone see how that worked? Yep. Okay. So, and then she points out, well, that worked in girls. Now, let's see what the story is. In boys, she said, all right, enough of modeling my brain and your brain with mine, and let's see if what we think is true. So now we're going to take this idea that it's androgens, go back to males, and see what we can do in males. All right. And so she wonders if androgens are responsible for masculinizing his song system. And so what we're going to do is now we're going to go back and block androgens in male development and see what's going to happen with that drug flutamide. Okay, once again, it grabs onto the androgen receptor and keeps it from working. And when I initially thought about this idea, I wasn't very happy because people had already given flutamide to day 1 zebrafinch babies. There's a guy by the name of Dr. Schlinger. Have any of you ever met this guy? All right, so he'd already done that when we were postdocs together, and I also did it in another study, and it didn't work. So I wasn't real hot for this idea initially, and I didn't do anything about it. And then there's this guy in Germany, Gar, who published this paper, and he showed that in the song system at least, the androgen receptors don't come up until day 9 post-hatch. And we had given the treatment back on day 1. So I went, oh, we missed the critical period. All right, it's up there in day 9. That's when I should have been given the flutamide. All right, so I designed this study. I was going to catch that window this time. All right, so what I did was I got one group that got flutamide, and I made sure they stayed on monstrous doses of this drug for their entire life, so their song system could not play catch-up later. Okay, so I didn't want the flutamide to like wear off and then some androgens then run in there and masculinize it. So they got 400 micrograms. Controls got implants at the same times. They got 400 micrograms from the get-go on day 7, so I had lots of flutamide already present in the song system when the androgen receptors developed. I kept them on very large doses, and we finally got tired of implanting them, so day 60 we took their brains. All right. It's a lot of work to keep implanting these guys over and over and over and over and being nice to them and stuff. So he's going to brag about this study. So one of the things you want to know is, well, does this flutamide drug even work in these zebrafinches? Of course, the androgen receptor in mammals is different a little bit from the androgen receptor in birds. So does this drug bind to the androgen receptor and have effects on the androgen receptor in these birds? That's one thing you want to check. Okay, this is what's called, at least in psychology, a manipulation check. So I want to see if it worked, and so we looked at the weight of the testes, and the really good news, testes of course are androgen-dependent tissues, and the flutamide really whacked the testes. So this is their weight in grams, and relative to controls, they only weighed about half as much. So this establishes that this drug can really have an effect on androgen-sensitive tissues, all right? And we're giving it at big enough doses that if it can, we would expect if it's going to have an effect, it should. All right, does that make sense? All right, good news. It works in birds. At least their androgen receptors aren't so different. So he's going to go ahead and puff this study a little bit, and he said, all right, look at this nice effect on the number of RA neurons. Remember, that's one of those sexually dimorphic traits. And if we look at the number of neurons in RA from controls relative to the flutamide treated, we see, yes, there is a significant reduction, p less than 0.05. Is that a criterion with which you're familiar? Okay, so we'll talk a little bit more in that in lab. So yeah, I got a significant effect on the number of RA neurons. Life is good. Almost. Okay, so males have about 15,000 neurons in RA, and if I'd put a control females in here, they have about 4,000 RA neurons. So did it sex reverse them? No, not even close. Okay. So as she points out, well, that effect was kind of small. And we were hypothesizing that androgens were responsible for masculinizing this whole system. She asked, did flutamide have any other effect on any other aspect of the song system? And he's embarrassed to admit, no, it didn't. Oh, not good news for my whole hypothesis, right? So it's not looking so good that androgens can masculinize this song system, right? Not good at all. So what we come away with on that study is flutamide can have a profound effect on androgen-sensitive tissue, really had a big effect on the testes, but they're probably not really important for masculine development of the song system, at least from day 70 to 60. They may fine-tune it a little bit, but they're not the big triggering factor. Doesn't look like it. Does that make sense? Alas, androgen's not so good. We got a nice paper out of it anyway. So Sun-Hee Park and Jen Shaw, Carolyn Kim are all off in medical school. So is Mike Lung. He never told me where, though. It's another sex difference. Men don't communicate as well as females. All right, Linda Kim is still on this planet, last I knew. Okay, so now she's going to say, maybe we should go back and look at this estrogen hypothesis again. And what if you blocked the estrogen synthesis in developing males? So we know if we give estrogens to the little girls, their song system will masculinize, right? At least in part. So what if it's estrogen? Let's go back and take that idea back to little boys and see, is estrogen responsible? So a colleague of mine, Julie Wade, did this study, and what she's going to do with this study is she's going to give a drug called Fadrazole, which I'll have up in another slide. But what that drug does is it blocks this enzyme aromatase, okay? So it's going to block the formation of estrogens from androgens. So the drug aromatase is going to block this enzyme that allows androgens to be turned into estrogens. By the way, Fadrazole I don't think is used clinically, but there are some very similar molecules that are. So there's a drug called Arimidex that's used for estrogen-sensitive breast cancers. The same idea. It blocks that aromatase enzyme so the body can't be making estrogens. All right, so my colleague blocked this aromatase enzyme, and here's some data when she did it injecting into eggs, looking very early in life. And what you can see here in this slide is that if you look at Fadrazole treated, that's the drug that blocks the aromatase. It's the same as the guys that got saline. So it didn't do anything to the song system in RA or in HVC or in area X. So blocking the formations of estrogen in males didn't do anything. All right, believe me, this really rocked us back. We were very surprised. This time she did it in eggs. She also did it post-hatch. She could never get this song system to change very much with this manipulation. Okay, and she was able to establish this manipulation would actually block other aspects of feminine development. So instead of developing ovaries, they would develop this thing that looked like an ovotestis. It would actually be, because the ovaries in birds need some estrogens to develop. And if it's not there, they'll go flipping back to look like a testis. So her birds were developing this ovotestis, kind of a half-half kind of thing. So she could show the drug was working in these animals, but not in the brain. Okay. Ooh, bad news for the estrogen hypothesis, even though it looks so good. All right. I loved this dogma, and I was desperate to save it. Okay, and so one of the things that also came out of her paper was that this enzyme, although it shuts down most of estrogen synthesis, it doesn't completely shut it down. So if we look at this graph, this is the estrogen production under saline and under fadrazole, and yeah, it significantly reduces it, but it doesn't make it go to 0 levels. All right? So what I thought was, hey, if one could, maybe the song system is exquisitely sensitive to estrogen, and just a little bit of estrogen is enough to kick off that development. All right? And so the little bit that's left is still kicking off development. Maybe that's what's going on. So what I decided to do was look at a dose-response study of estradiol, and that's what you're replicating in lab. All right. So we wanted to gauge the sensitivity of the song system to estradiol, see if it's really sensitive to estradiol. Can just a little bit kick off this masculine development? And also to see if different aspects of the song system were differentially sensitive, and is there like a threshold effect, or is the response to estradiol graded? So this ought to be pretty familiar to you. This is at least some of the goals for your lab. Okay? And we did publish this not so long ago. Janet Lynn, Sun Hee, and Jen Mankowski helped me on that. They were undergrads that were helping. And when I did it, I had 4 groups. I didn't have any males like you guys have. Okay? So for this, we added a group of males for you, but I had 4 groups. They either got 0, 5, 15, or 50 micrograms of estradiol, implanted them on day 1, sacked them on day 100. So again, what I was desperate to do was save this hypothesis, seeing, I was hoping that the song system would be exquisitely sensitive to estradiol, and then we could say, yep, the dogma was true. Here's how it came out, at least in my hands. Don't know about your hands yet. So here's HBC volume. Here's my different treatment groups, the different doses of estradiol. And this is what I had to conclude from my data. Okay, when I gave them 5 micrograms in that group, by the way, each bar here represents a different animal. It didn't masculinize them at all. All right, they were just like the 0 controls. And here, again, you can see these asterisks, 50 and 15 micrograms. These groups significantly masculinized relative to controls. But 5 micrograms didn't do the job. So you might say, well, 5 micrograms doesn't sound like a whole lot. But if you give 5 micrograms of estradiol to a baby rat pup who's a lot bigger than a baby zebrafinch, you can completely change her brain, masculinize her brain for the rest of her life. So when she grows up, she won't have an estrous cycle, okay? So her brain won't be able to cycle an ovary like a regular female brain will, okay? So 5 micrograms, at least in other systems, is like a monster dose, okay? Here, it didn't do squat. All right, so is it exquisitely sensitive to estradiol? No, okay. This is bad news, real bad news for the dogma, right? Things are just falling apart right and left on this. Okay. The other thing that was in the literature was that you had to have at least 50 micrograms to masculinize, and my data showed that you don't need that much, but it's not exquisitely sensitive. All right, so let's go now and look at some problems with this gonadal steroid story and the zebrafinch song system. The first one was estradiol only half masculinized the song system at best. Okay, that alone wasn't great, but we start putting it together. We blocked androgen receptors in males, didn't do very much. Okay, so it doesn't look like it's androgens. We inhibit estrogen synthesis in males, didn't touch it. Okay, I was desperate, and we could have saved that. The dogma, if the song system is exquisitely sensitive to estradiol, and the study I did, it doesn't seem exquisitely sensitive to estradiol, all right? So this whole dogma, hormonal dogma, is kind of unraveling, okay? I didn't like this. This is what I grew up believing, okay? So we had some thoughts, all right. So there's always the idea that the timing or duration or the dose of your treatments is wrong, and if you just change the timing or you change the duration, maybe you give something pre-hatch and post-hatch, you'll get some bigger effects, or the dose of your treatments need to be bigger. That's always a possibility and one that we could consider. There's some things that argue against that. The fact that we're seeing effects on other tissues suggests the doses are okay. And the timing, there's always possibly a problem. But at any rate, we were starting to have some heretical thoughts. And one heretical thought was that maybe sexual differentiation of the song system is determined by something other than gonadal steroids. Could there be somehow differences in the male and female genome that are kicking off these things that aren't dependent on steroid hormones? Okay? So let me talk a little bit about the sex chromosomes in birds. Birds don't have X's and Y's, okay? The male is a double Z and the female is a ZW. And I used to think these were just different names for X's and Y's, but it turns out if you look at the genes, these chromosomes are not homologous to X's and Y's. They're completely different sets of genes. So the male, again, is ZZ, and the female is a heterozygote ZW. So hang on to that. So to test if it's something about this genome between males and females that's not steroid dependent, or it's steroid dependent masculization, that somehow we've missed the timing or duration. What we'd really like to do is get a bird like this, okay? A bird that is male on one half and has the male genotype on one half, the ZZ on one half, and the female ZW on the other half, okay? And then that brain would be sitting there in the same head, so it'd see the same set of hormones at the same time, okay? But we'd have half the brain be one genotype and half the brain be the other genotype, right? That would be the perfect critical test between hormones versus genotype, right? Wouldn't that work? Wouldn't that be lovely? This bird lived. This bird lived, and this is the bird that drove me to hormonal heresy, okay? This bird was an experiment of nature. We did not create this in a lab. Nature created this thing, and it was a true hermaphrodite. It had male plumage on one side of its body, just like you see. This is not a Photoshop morph, by the way. This is a real picture. It had male plumage on this side of its body. It had female plumage on this side of its body. That's what drew it to somebody's attention at Rockefeller University, and they sent it to us. And it sat around in Dr. Arnold's office for many years, a couple of years, while we tried to figure out what the heck to do with this thing, because it's an N of 1, but it's very valuable. So finally he decided, all right, we got to open this up and see what it's like. So we open it up, look at its gonads, and we see, yes, it's a true hermaphrodite. It had both an ovary. Here you can see the egg follicles developing, and it also had a testis. Yes. Did it sing? Yes, it did sing, as a matter of fact. It courted females, as a matter of fact, and it tried to copulate, but it seemed to be sterile, and this kind of explains why. This testis doesn't look great as testes go because it's been exposed to some estrogen. And if you do that, it kind of messes up testes. All right. So, but it did have a testis on the male plumage side. It had an ovary on the female plumage side. Whoa. All right. So it's a true hermaphrodite. And so then the question became, what about the brain? What about the brain? And so we had a marker for the W chromosome, and we found that this W chromosome that determines the female was only expressed on half of the brain, almost split spot down the middle. It's amazing. And it, of course, was on the side that had the female plumage and the ovary. Okay, this side had a ZZ genotype. So genotype split right down the middle on this guy. So just to review, if it's hormones that are driving the sexual differentiation of this system, then what we ought to see, hormones should be acting on both sides of the brain, and it ought to be symmetric, right? If there's something about genotype that's going to be, genotype will act differentially on these two sides, and it will be asymmetrically developed. Does that make sense? This guy's going to, he, she, it is going to do it for us, because only on this side of her brain does she have this it has this genotype, okay? So if there's something about having this genotype, the neurons having this genotype that's driving development, then we're going to see a difference between this side and this side, okay, if it's the genes, okay? If it's the hormones, this is the same brain, should be bathed in the same hormones at the same time, okay? So the development should be symmetric, okay? And that's the dogma, that it should be symmetric, right? Okay, everybody see where this is going? All right, and now the answer. The genetically male side was more masculine than the female side. So is it genes? Yes. Okay, so something that's not steroidal is driving this development, at least in part. And so believe me, I was the guy who drew these brain areas. They gave me this brain and they didn't tell me which side was which. So I was blind. And after I drew this, I just sat back and I went, whoa, this thing's asymmetric. This is way asymmetric. That could be important. So then I actually was lucky enough that I had kept all my records of all my males, left versus right, so I could have some sense of how asymmetric our brains usually. And they're down in this range, and this guy who is half-half is up here. So it's way asymmetric than everybody else. Okay, so that's again evidence that this is way out of the norm and suggests that yes, genes are responsible. Something about the genotype is responsible for driving the sexual differentiation. All right, so that's what led me into heresy. So then I have to admit, sexual differentiation of the brain is not exclusively driven by gonadal steroids. Gonadal steroids can still play a role, but they aren't the whole story, okay? And I very reluctantly came to this conclusion, but that bird forces me to do that. All right, everybody agree? Excellent. This, by the way, hasn't hit the textbooks yet, but it will. So we got a really nice, this is the paper I'm probably proudest of. This one ended up in PNAS. Do you all know about journals? Okay, so PNAS, good or bad? Way good. Okay, so some people say there's Nature, Science, and then PNAS in all of science. So we managed to get this one in PNAS. Even if I'm second author, I still feel pretty good about that one. All right, so going back to your study. So what you're going to do is you're essentially going to try to replicate that dose response study. So you're going to try to, first of all, you're going to try and see if we can get sex differences, all right? And by the way, so far, as I said, your data look great. We're going to gauge the sensitivity of the female song system to estradiol and see if masculinization is an all or none event. I'll talk more about this some lecture next week. We're going to see if the different parts of the song system show differential sensitivity to estradiol. By the way, how many dependent variables do you have? Okay. Dependent variable is what you are what? Measuring. What are you measuring? Area X? HBC? All right, so how many do you have? It's a trick question. 3. Okay, so you're going to, one of your things is you're going to see, oops, change slides here, is you're going to see if there's differential sensitivity to the different doses in those 3 dependent variables. Okay, and we're going to also be able to see, did any of our doses fully sex reverse? I didn't have males back in my study, I couldn't say, but you will be able to. All right, So we'll get you out of here a little bit early today. Gonadal steroids are not exclusively responsible for sexual differentiation, at least in the zebrafinch song system, maybe even in mammals. And genetic differences between the sexes are partially responsible for sexual differentiation. Okay, see you next week.